Good morning, students. We will continue our topic carbohydrate metabolism. Today's competency is competency number 3.6. In this competency, the topic is TCA cycle, its pathway, energetics, regulation, and its amphibolic nature. So, TCA cycle, the long form is tricarboxylic acid cycle. This is so called because the carboxylic acids form as an intermediate of this cycle are having three carboxyl groups. Hence it is called as tricarboxylic acid cycle. It was discovered by the scientist Sir Hans Krebs. Therefore it is also called as Krebs cycle. The first product which is formed in this cycle is citric acid. Therefore another name of TCA cycle is citric acid cycle. So site, this cycle occurs in mitochondria of all cells of the human body. It is the main cycle which provides ATP to our body. So this is the TCA cycle pathway. In the first step, the 4-carbon oxaloacetate combines with 2-carbon acetyl-CoA to form 6-carbon citric acid, which is the first compound in this pathway. The enzyme required for this step is citrate synthase. Here coenzyme A from acetyl-CoA is removed. Now this citric acid undergoes dehydration to form cis-aconitate, which is a unstable compound which readily combines with water to form isocitrate. Both these reactions are catalyzed by the same enzyme aconitase. Now isocitrate dehydrogenase undergoes oxidation by removal of 2 hydrogen atoms with the help of enzyme isocitrate dehydrogenase. Now these 2 hydrogen atoms are accepted by NAD and get reduced to NADH. Now the oxalosuccinate which is formed now undergoes decarboxylation with the help of same enzyme isocitrate dehydrogenase to form alpha ketoglutarate having 5 carbon. Now this alpha ketoglutarate undergoes simultaneous oxidation and decarboxylation by removal of 2 hydrogen atoms and 1 carbon in the form of carbon dioxide with the help of enzyme alpha ketoglutarate dehydrogenase, and the product form is succinyl-CoA. Here coenzyme A is added in the reaction. The 2 hydrogen atoms which are removed during oxidation steps are accepted by NAD to form reduced NADH. Now the 4-carbon succinyl-CoA which is formed is a high energy compound which is broken down with the help of enzyme succinate thiokinase, and coenzyme A is removed. In this step, large amount of energy is released. That energy is used for the phosphorylation of GDP to GTP. So this is an example of substrate level phosphorylation in TCA cycle. Now the succinate or succinic acid which is formed is then undergoes further oxidation by removal of 2 hydrogen atoms to form fumarate. The 2 hydrogen atoms removed during the reaction are accepted by a coenzyme FAD which get reduced to FADH2. The enzyme required for this step is succinate dehydrogenase. Now the fumarate or fumaric acid which is formed undergoes hydration with the help of enzyme fumarase by adding water molecule to form malic acid or malate. Now this malate undergoes fourth oxidation in this cycle to form oxaloacetate. Here two hydrogen atoms are removed and again these two hydrogen atoms are accepted by a coenzyme NAD, which is reduced to NADH. The enzyme required for this step is malate dehydrogenase. Now in this way the oxaloacetate, which is the substrate of this TCA cycle, is regenerated at the end of the pathway. So in this pathway acetyl CoA is completely utilized by removing two hydrogen atoms at two different steps, and oxaloacetate is regenerated as it is. So this is TCA cycle pathway. Now come to the energetics of TCA cycle. TCA cycle gives 10 ATP molecules. In the third step, the isocitrate dehydrogenase removes two hydrogen atoms from the substrate and which are accepted by NAD to form NADH, and that NADH, when enters into the electron transport chain, gives 2.5 ATP molecules. Similarly, alpha ketoglutarate dehydrogenase also removes two hydrogen atoms from the substrate which are accepted by NAD to form NADH, which also gives 2.5 ATP molecules. In the fifth step, substrate level phosphorylation takes place. The enzyme required for this step is thiokinase succinase. 1 GTP is synthesized, which is equivalent to 1 ATP. In the sixth step, again two hydrogen atoms are removed by the enzyme succinate dehydrogenase, which are accepted by FADH to form FADH2. This FADH2, when enters into the electron transport chain, gives 1.5 molecules of ATPs. Lastly, in the eighth step, malate dehydrogenase causes oxidation of substrate by removing two hydrogen atoms which are accepted by NAD to form NADH, and this NADH also gives 2.5 ATP molecules in the electron transport chain. So in this way, in one cycle of TCA, 10 ATP molecules are formed, and this energetics is given according to the new values of NADH and FADH2 given in the book. Previously in the book they have given that NADH gives 3 molecules and FADH2 gives 2 ATP molecules. After energetics, come to the allosteric regulation of TCA cycle. The three enzymes of TCA cycle are important. First is citrate synthase, second isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase. Citrate synthase is inhibited by ATP molecules. The main purpose of TCA cycle is to generate ATPs. When the concentration of ATP inside the cell increases or inside the mitochondria increases, then this ATP act as a allosteric inhibitor of this enzyme citrate synthase and decreases the TCA cycle pathway. Next, isocitrate dehydrogenase is also inhibited by NADH molecule. So this NADH ultimately gives ATP molecules in the electron transport chain. When the concentration of NADH increases, they inhibits the enzyme isocitrate dehydrogenase. But when the concentration of ADP increases, which indicate that cell requires more ATPs. When concentration of ADP increases means there is deficiency of ATP. So this ADP act as a allosteric stimulator of this enzyme isocitrate dehydrogenase and increases the rate of TCA cycle. Succinyl CoA and NADH act as a allosteric inhibitor of the enzyme alpha ketoglutarate dehydrogenase. So this is about the allosteric regulation of TCA cycle. TCA cycle is mainly for synthesis of ATP. So our next factor which regulate TCA cycle is availability of ATP inside the cell. When the energy level of the cell is low, then the TCA cycle is stimulated and it operates in faster rate. This TCA cycle is also closely related to electron transport chain or respiratory chain. The next factor which regulate the TCA cycle is availability of oxygen. When there is deficiency of oxygen, that is anaerobic condition persists in the cell, which inhibits electron transport chain. Therefore, NADH and FADH2, which are utilized in electron transport chain, are accumulated, and hence there is inhibition of TCA cycle takes place. So deficiency of oxygen not only inhibits the electron transport chain, but it also inhibits the TCA cycle through this mechanism. So this is about the regulation of TCA cycle. Now we will see the amphipolic nature of TCA cycle. In the metabolism, up till now we have studied various pathways, and we found that some pathways are catabolic and some pathways are anabolic. But TCA cycle is both catabolic as well as anabolic. Therefore, it is called as amphipolic in nature. First we will see anabolic nature of TCA cycle. The intermediates of TCA cycle are used for the synthesis of various substances. Therefore, you can say TCA cycle is anabolic in nature. For example, acetyl CoA is used for fatty acid synthesis. Alpha ketoglutarate is transaminated to form glutamic acid, which on decarboxylation form gamma amino butyric acid, which is a neurotransmitter. Succinyl CoA is used for heme synthesis. Succinyl CoA is also used for synthesis of ketone bodies. Malic acid is used for synthesis of glucose by gluconeogenesis pathway. Similarly, oxaloacetate on transamination gives aspartic acid. So the intermediates of TCA cycle are used for synthesis of various substances, and this is the anabolic nature of TCA cycle. Catabolic nature of TCA cycle is, we have seen in the pathway, acetyl CoA is completely oxidized and it is converted into carbon dioxide and hydrogen atoms. This is an example of catabolic nature of TCA cycle. Now we will see the anaplerotic nature of TCA cycle. Anaplerotic nature means there is continuous influx, means adding into, and continuous efflux, that means removal from the TCA cycle. This means that the intermediates are continuously added from various sources in the TCA cycle, and as body required, the intermediates of the TCA cycle are removed for synthesis of various compounds. So this is called as anaplerotic nature of TCA cycle. Sometimes TCA cycle is also called as metabolic traffic circle. So these are the examples of influx in the TCA cycle. Glucose on gluconeogenesis get converted into pyruvate, or alanine on transamination get converted into pyruvate. This pyruvate is converted into acetyl CoA, and this acetyl CoA enters into the TCA cycle. Similarly, the amino acids like histidine, arginine and proline, they get converted into glutamic acid, and this glutamic acid later on get converted into alpha ketoglutarate by deamination. The amino acid like valine, isoleucine and methionine, they get converted into propionyl CoA, and this propionyl CoA undergoes carboxylation reaction to form succinyl CoA. So various intermediates are synthesized from different amino acids, and they join the TCA cycle in between. Similarly, phenylalanine and tyrosine, they get converted into fumaric acid and enters into the TCA cycle. Aspartate also joins the TCA cycle by converting into oxaloacetate. So these are the examples of influx in the TCA cycle. That means various substances, here amino acids, get converted into different intermediate compounds and they enter into the TCA cycle. So TCA cycle may starts from any intermediate. So this is influx in TCA cycle. Efflux or removal from TCA cycle, the examples are here. The acetyl CoA are used for fatty acid synthesis. Alpha ketoglutarate, which is converted into glutamic acid by transamination, which on decarboxylation get converted into gamma aminobutyric acid, which is a neurotransmitter. Similarly, succinyl CoA is removed from TCA cycle and used for heme synthesis, as well as succinyl CoA is used for synthesis of ketone bodies. Malate is used for synthesis of glucose by gluconeogenesis pathway. Oxaloacetate from TCA cycle is removed and used for synthesis of aspartic acid. So these are the examples of efflux from TCA cycle. Efflux means removing the intermediate and converting them into various compounds. So we have studied influx of TCA cycle as well as efflux from TCA cycle. Such nature of TCA cycle is called as anaplerotic nature of this cycle. And last, we will study the common poisons which inhibits the TCA cycle. So there are 3 poisons which inhibits the TCA cycle. First is fluoroacetate. Now this fluoroacetate inhibits the enzyme aconitase. Due to this inhibition, the citrate is not converted into aconitate, and this type of inhibition is non-competitive, irreversible non-competitive type of inhibition. Now the next poison is arsenite. It inhibits the enzyme alpha ketoglutarate dehydrogenase, and this inhibits the reaction in which conversion of alpha ketoglutarate to succinyl CoA occurs. The mechanism of inhibition is again by irreversible non-competitive inhibition. The third enzyme is malonate, which inhibits the enzyme succinate dehydrogenase of TCA cycle, due to which the conversion of succinate to fumarate is affected, and this type of inhibition is competitive type of inhibition. So this is about the TCA cycle, its regulation, energetics its amphibolic nature, its anaplerotic nature, and its inhibition. Thank you.